Hello everyone, I hope you are all doing well. My name is Jose Peña and I am a master student in biological science at Pontificia Universidad Javeriana. I'm excited to have the chance to share with you today our work on triple negative breast cancer titled Immunological and Transcriptomic Alterations in the Tumor Microenvironment of Triple Negative Breast Cancer and a sTILs-based approach. To begin, I would like to talk to you about breast cancer. Breast cancer is the most common malignant neoplasm among women worldwide. Triple negative breast cancer is associated with an aggressive phenotype and lower survival rates. Recent studies have shown that the cholesterol within the tumor microenvironment plays a crucial role in the disease development and progression, particularly the infiltration of the immune system. Evidence suggests that a higher level of stromal tumor infiltrant lymphocytes within the tumor microenvironment is a marker of favorable clinical response, which has led to the immune status of the tumor being considered a key prognostic factor of the disease. For this reason, our study aims to explore the immune component in Colombian patients with TNBC. Specifically, our objective is to evaluate the immune cell distribution and transcriptomic profile in the tumor microenvironment of patients with non-metastatic triple-negative breast cancer according to ST levels. For this purpose, in this study, 71 untreated tumor samples from patients with a primary diagnosis of TNBC were analyzed. STIL infiltration was assessed by a pathologist on hematoxylin and eosin stain slides, and RNA was extracted from formalin-fixed paraffin-embedded tissue blocks for RNA-seq. Immune cell infiltration was estimated from a TPN normalized gene count matrix using Excel and CyberSort. Immunological subtypes were identified through Cummings clustering. Then, Spearman correlation analysis were used to compare ST levels with immune infiltration estimates. Overall survival was also evaluated across the identified cluster. And finally, DEX were identified with DESEC2 followed by enrichment analysis. All analyses were performed in RStudio software. We identify in our results that according to ST levels, patients were classified into two groups, low STIL and high STILs. Our analysis revealed a positive correlation between STIL percentage and the immune score obtained by Excel. However, the absolute score showed no significant correlation with STIL or the immune score. Despite this, both the convolution methods were used to estimate immune clusters. Our results related to the clustering analysis revealed that Cummings clusterings identified two optimal clusters based on Excel results. The optimal number of clusters was determined to be two using the gap statistic, which compares total within cluster variation. Additionally, the silhouette plot analysis showed that most sample has valued close to 1, indicating a strong clustering assignment. Perhaps it is difficult to see the details in this figure, but I encourage you to take a closer look at the full poster. Here we can see the immune clusters formed using Cummings analysis. Similarly, two clusters were identified through CyberSort analysis. In both methodologies, patients with high STILs were predominantly grouped into Cluster 1. Cluster 1 showed a higher immune score and absolute score than Cluster 2, so Cluster 1 was designed as the immune enriched and Cluster 2 as immune low cluster. The cellular composition in each identified cluster is represented in these heat maps, showing the Excel and CyberSort results. The significantly different cell types between clusters are shown in bar charts. According to Excel, the immune-enriched cluster exhibit as dual behavior, with an increase in antitumor cells like CD4 T cells, CD8 T cells, NK cells, macrophages, and M1 macrophages, as well as protumor cells like Tregs and M2 macrophages. This demonstrates that while these individuals have higher immune infiltration in their tumor microenvironment, it is not solely associated with antitumor immune cells. Meanwhile, CyberSol results support this trend, showing increased antitumor cells, though Tregs were absent. An increase in other cell subpopulations, such as M0 macrophages and memory B cells, was observed. However, it is important to note that cluster formation based on cyber results was not as robust as observed with Excel, though the cell pattern was similar. Once we established the immune differences between both clusters, we conducted a transcriptomic approach to identify differentially expressed genes between both conditions. Using the SEC2, we identified 4 decks between the immune low and immune enriched cluster, with 21 overexpressed and 18 underexpressed genes. Enrichment analysis indicated that the altered gene expression patterns were associated with changes in metabolic process such as arachidonic acid metabolism, which it is known to affect immune regulation. Finally, to assess the impact of immune cluster on prognosis, we evaluated 5-year overall survival between immune clusters. While statistical significant differences were not observed, there was a trend indicating higher overall survival rates in the immune-enriched cluster compared to the immune-low cluster. This suggests a potential prognostic implication of immune cluster in TNBC patients. To conclude, I would like to emphasize that our study identified two optimal immune clusters for classifying TNBC patients. These clusters show differences in cellular composition, gene expression, and survival patterns. However, we want to highlight the variation in cell estimates obtained through both the convolution methods, emphasizing the need to increase sample size and conduct more in-depth analysis to refine immune grouping. Thank you very much for your attention, and I wish you all a lovely day.